Good morning, everybody, and welcome to the session of sterilization. So, in today's session, what are we going to discuss? That is the definitions of sterilization and disinfection, something about CSSD. Then we will start with the methods of sterilization, which we will continue in the coming videos as well. So, let's proceed with the definitions first of all. So, what is sterilization? Sterilization by definition is the removal or destruction of all the microorganisms, including spores. That is important for you to remember. It must include the spores also, including spores. And how will you remember that it is including spores? So S is for sterilization. The S is for sterilization and S is for spores here. S is for spores. So it includes the spores. If we compare with the definition of disinfection, then in disinfection it is the removal of the most of the organisms, but not the spores, but not spores, not the spores. Also, one more difference is that disinfection is used for inanimate objects and not for body surfaces. It is used for inanimate objects and not for the body surfaces. Okay. So this is the definition of sterilization and disinfection, and further we proceed with the methods of sterilization. Before that, just a little bit about the CSSD. So what is CSSD? So C stands for the central, S stands for sterile, another S stands for supply, and the D stands for department. So it is the Central Sterile Supply Department. It means it is supplying all the sterilized instruments, linen, etc. to the whole hospital. It is the department where all the sterilization takes place. And then the thing is how we are dividing the department into what are the various areas in that department. So whenever we receive anything from the anywhere from wards, OTs, or anywhere from the hospital, any staff, we receive it at a counter which is also known as the receiving counter. It is called the receiving counter. So from receiving counter, the products are then sent to an area which is designated as the decontamination area. And what are we doing in that decontamination area? So here we are washing the stuff with the soap or detergent to decontaminate it first of all. And after it is decontaminated, then what we are doing, we are sending that instruments or linen etc. to the second area that is known as, first area was the receiving counter, second is the decontamination, third area is the packaging area. So here the materials are packaged so that it allows, in a way it allows the sterilant to penetrate the the packaging material. So that is the purpose of the packaging. And after packaging is being done, now we are finally sterilizing it in an area called sterilization area. Excuse me. So what is happening in the sterilization area? Finally sterilization is happening. Mainly two methods are used. Either it is ET or ethylene oxide, or it is the autoclave. These two methods are usually used for sterilization in the sterilization area. And once everything is sterilized, we are now putting it in the storage area. Fifth one is the storage area, and after being stored, then as per the requirement, we are issuing it to the various areas. So next we have the issue counter. Here the things are issued and they are being then sent to various areas in the hospital, like it could be sent to OTs, it could be sent to wards, or wherever the requirement is. Okay, OT, wards, etc. etc. So this is how this works, and one very important thing to remember is that if you see, we are receiving at the one end, that is the receiving counter, and then we are going this way, right? We are going this way. We are not coming back, okay? So this is unidirectional. It is unidirectional so as to prevent any contamination. The things are not coming this way anywhere. It is just going in a one direction to prevent contamination. So this is all about the central sterile supply department. And after this, now we will start with the methods of the sterilization. Now in the methods of sterilization, we have physical methods or we have the chemical methods. Okay, we will discuss here the physical method first. So in physical method, we have number one, heat, we have radiation, and we have filtration. Three physical methods of sterilization. And in the heat method, further we have dry heat. We have either the dry heat or we have the moist heat. Now coming to the mechanism of action. What is the mechanism of action? Mechanism of action. So in the dry heat, first of all, the mechanism of action is denaturation of proteins. Denaturation of proteins. After this, we have electrolyte imbalance. We have electrolyte imbalance, and the third mechanism of action is there is oxidative damage. There is oxidative damage. In the moist heat, again we have the denaturation of the proteins. We have denaturation of the proteins and we have oxidative damage. Only electrolyte imbalance is not there. So what are the mechanism of action in moist heat? We have denaturation of proteins, and we have the oxidative damage. These are the two mechanisms of action in moist heat. So we have in totality physical and chemical methods. Chemical methods I will take up separately. In physical method we have heat, radiation and filtration. In heat we further have dry heat and moist heat, and we have discussed the mechanism of action. After this we will come to the methods of dry heat now. Okay, we will take up one by one. So in the first method of dry heat, we have flaming. So what is happening in flaming? This is the flame you can see, and this is the burner which is known as the Bunsen burner. Bunsen burner having a flame. You can see the flame. So what are the temperatures at which this flaming is done? The flaming is done at a temperature, outermost temperature, that is the 800 degrees Celsius. Then we have the middle temperature of 1200 degrees Celsius, and the innermost layer of the flame, that is having the temperature of 400 degrees Celsius. So basically what is happening, you can see the flame. This is how the flame looks like. It has outer, middle and inner layer. Outer layer of the flame, then there is a middle layer of the flame, and then there is the inner layer of the flame. So these three temperatures of the outer, middle and inner layer, these are being told here, 800 degrees, 1200 degrees and 400 degrees Celsius respectively. Now what are the uses? If we talk about the uses of flaming, what is the use? So it is being seen in this diagram itself that it is holding an inoculating loop. This is the inoculating loop. So the most common use is sterilizing inoculating loop. That is the use of flaming. The next method is incineration. Next method of dry heat, that is incineration. So incineration means what? Incineration means burning to ashes. Burning to ashes where the volume is decreased. where the volume is decreased by 80 to 85%. Okay. The equipment in which incineration is done, that equipment is called incinerator. Incinerator. Okay. And now what is the temperature? That is important for us to remember. What is the temperature at which this incineration is working? That is the 1000 degrees Celsius to 1200 degrees Celsius. Okay, so this is about the incineration. Next method what we are having in the dry heat, that is the hot air oven. It is another method of dry heat. So what is happening in hot air oven? This is the equipment which you can see. This is the hot air oven. It has racks. It has these racks where anything to be sterilized is put up, and then we have a thermometer over here to record the temperature. We have a thermometer over here which records the temperature, and this is the knob. You can see this is the knob which is used to adjust the temperature. Okay, this is called the hot air oven. So what temperature, that is important for us to remember at what temperature this hot air oven is working. It is working at 160 degrees Celsius for 2 hours. And what are the equipments, what are the things which are sterilized in this? What are the uses of hot air oven? So it is used to sterilize anything that is having grease, dust powder, glassware. That is important for us. Glassware, including the sharp instruments. Then glove powder. We have gloves powder. Okay. What type of glassware can we use? What type of glassware we can use? So in glassware we can use pipette. They can be used to sterilize pipette, flask, test tubes, etc. Right? This is basically about the hot air oven and its uses. Now there are certain controls. There are certain controls which are used to see the efficiency or functioning of the sterilization happening in the hot air oven, whether it is being functioning properly or not. And these controls are further divided into physical, chemical, and biological controls. So what is the physical control? I have already told you we have a thermometer in this. So in physical, a thermometer is there to see if the proper temperature is being maintained or not. Then we have various chemical indicators also. These are controls or also known as indicators. We have chemical indicators also, we will take up in the next video. And we have biological controls or biological indicators. As the name suggests, biological, so it is using microorganisms. Which microorganism is used here? So here it is using the spores of Bacillus atrophius. Spores of Bacillus atrophius. This is used as a biological, very important to remember. Spores of Bacillus atrophius is used as a biological indicator of hot air oven. So this was all about the methods of dry heat in the sterilization. In the next video, we will take up the moist heat method. So please continue watching the videos for further methods of sterilization. Thank you.